literature review will collect. Then only will come to methodology. First, objectives has to be fixed. And preliminary data, we should have some preliminary data that is called your proof of concept. Then, for that, with that also some statement limitations are there. Finally, we can go for the conclusion session. One thing comes to my mind this juncture. During 1992, I went to National Lady Harding Medical College for a workshop on group-based streptococcal infection. That was Lady Harding Medical College is supposed to be the regional center for advanced studies in streptococcal diseases. That was identified by WHO. Dr. Kunti Prakash, she was the former director, head department of microbiology. I met her and when I met her, she was telling the preparation of methodology or objectives for a PhD thesis should be the first. When I approached her for my thesis, at the time I got registered for my PhD, she asked me to give one-page material, one-page idea, and 5 objectives with 1 page and 5 objectives. It was very difficult for me to prepare that way. I gave her about 10 pages. The next day, she just gave it to me and said, I asked you to prepare 1 page and 5 objectives. Next day, I prepared about 10 objectives and 1 page material. She told me still you can prepare only 5 objectives. Then I corrected it. It took me another 1 week time to prepare it. But I understood when I gone through all the literatures, to preparing 1 page material, it's very difficult. A 10-page material today, it's easy to prepare. So her idea, still I follow. That was the So methodology in the sense, to put it in a nutshell, the methodology explains the procedures that we use to achieve the goal and objectives. Approach to the question, what comes to your mind, and also would have searched literature. Based on that, we will be approaching. We'll have to design, research subjects, then inclusion or exclusion criteria, sampling procedure, controls, comparison groups, data needs, analytic techniques, plan for interpreting results, and finally ethical issues. All this we need to consider before going for fixing your objectives as well as methodology. This is what we got some idea. In 1992-94, I went for a training on monoclonal antibodies to National Institute of Immunology. That time only I met Dr. Kunti Prakash also. He told me you need to have only 5 objectives for the PhD thesis. For monoclonal antibodies, it's easy. Antibodies are probes. It has got a wider application. At the time, 1985, Kobler and Milstein, they got Nobel Prize. Hence, I wanted to bring that technique to our laboratory. I went for a training on that. I understood that when I got the training, so we need to have infrastructure in our place. It's a growing. Our department was only, it was in a 2-3 years old department, 5 years old department. We are not having that much facility. Today we can able to do this very easily because we have an engaged institution. We have very good infrastructure, state-of-art facilities has come. One Dr. Ramanathan Lab and Institute of Advanced Studies and PG College of Arts and Science itself has got a cell culture facility today. At that time, there was no much facility was available. Even though I got trained when I came back, I was trained for 1 or 2 years. Dr. Appalraji was helping me to prepare antibodies with rabbit antibodies. Rabbit we used in PG Medical College at the time, using rabbits for the generation of antibodies. We wanted to have something alternative to rabbit antibodies. We have chosen chicken as a model. At the time, there was no much publications on chicken antibody. So later on, we understood the chickens can be used, and also we can purify the antibodies from eggs. Egg yolk antibodies can be used for many purposes. Now it's been established field today. We go about up to 20-30 years of my experience, I could understand. Globally, there are many industries has come, many antibodies has already available with chicken as a source of antibody. With that, we have worked about 20 theses on this, and we have proposed one advantage with the chicken antibodies. Antibodies is the general molecule which will be useful for the treatment of bacterial, fungal, viral, even cancer antigens, and also venom antigens. That is how we were able to understand this potential of this antibodies. So this is the general protocol, novel IgY antibody production and its applications. Today, a few years back, we have made this slide by understanding what is the potential of it. So when you immunize chicken, 21 weeks old white Leghorn chickens, it will maternally, it will be the antibodies will be raised. It will be transferred to the yolk after some time. For example, within 1.5 months, the antibodies slowly you will get it from eggs, collect the eggs and store it at 4 degrees centigrade for a period of 6 months, maybe. And you can keep on collecting. Each chicken you can get about around 250 to 300 eggs you will get. Roughly about 50 to 100 milligrams of antibody per egg yolk. It makes about 20 to 30 grams of antibody from one chicken. That is the potential of chicken antibodies, which can be replacing, these antibodies can replace mammalian antibodies. Nowadays, you can even treat viral diseases. There are some viral diseases, and it can able to replace some antibiotics. For example, poultry infection. Normally, antibiotics are used. There is a proposal you can replace the antibiotics with antibodies, so that the resistance of antibiotics we can able to control. So that way also we got in a project. These are all some of the slides which I made it, or each plate here mentioned, or one concept. Otherwise, it's a project, PhD thesis, or we have funded projects. First one is dental caries, Streptococcus mutans, which causes dental caries. We can prevent this using chicken antibodies, because the enormous amount of antibodies we can able to prepare. And preparing these antibodies, we can make some products like mouthwash, or you can incorporate into the gel or lozenges. With that, we can able to control. Next one is your antibodies. These antibodies can be used for treatment of snake bite. So already there are antibodies. Our antibodies are the antidote, or antivenom is available, which is being prepared from commercially from horses. That is the time, more than 100 years. This is the technique which we are using it. In India, we have now 4 important snakes like cobra, kite, and Russell viper, and sawscale viper. These big four snakes are poisonous snakes in India. Only these snakes bites only we need to treat them. Otherwise, there is no treatment required for any other snake bites. There are about more than hundreds types of snakes. Snakes are there. So only these four snakes has to be treated. So the one of poisonous, only these poisonous snakes are there. Only four poisonous snakes. Hence, the polyvalent antibodies, which is commercially available, has been used prepared from horses. This can be replaced with the chicken antibodies. That is the concept we have developed. And we have made a proposal. We got funding also for this. And it's almost in this industry, you'll be transferring this technology to industry. That's the stage we are. And third one, you have this is an antibody which we raised with chicken using beta-glucan chickens. This is against poultry infections. Antibodies are alternative to antibiotics. That is the concept we worked on. Poultry infection, mostly it is caused by salmonella. You have a special type of E. coli, Campylobacter. So we have taken 4 bacterial pathogens, we raised antibodies. We have raised antibodies, we tried with young chicks from day 1 old to 42 days. What is the changes like comparing with antibiotics, which they are commercially used and what they are using it? We were able to get good results that antibodies can be used instead of antibiotics. It is it's an SIBRI project. And so we have collaborated with these states. We are able to complete this project. The chicken can gain about half kgs of weight when you use yolk powder or spray dried powder with antibodies, which can able to, it's a functional food, which can able to increase the weight it can gain as well as it can able to neutralize any pathogens challenged like salmonella or Campylobacter. It's being transferred. This technology has been transferred to industry. Next one is like anti-dandruff. Dandruff is caused by Malthusia sulfur. We have raised antibodies against this also. It neutralizes the Malthusia sulfur. That's the concept we got in this. And next one is rabies. We got small funding from Tamil Nadu State Council for Science and Technology. We made a proof of concept. This project we have associated with or collaborated with Pasteur Institute of India. Now we are Dr. Ashok is working in PhD for his PhD. The concept is like we are raising antibodies against virus, rabies virus, G protein, and also available commercially available antigens, vaccines we have taken, and we are trying to raise the antibodies. We wanted to neutralize this. Neutralization studies we will be doing it in an animal model at Pasteur Institute. Similarly, for the infection caused in cattle, which is mostly Staphylococcus mastitis. Mastitis is about another big industry loss is there, about 6,000 crore industry loss is there due to the mastitis alone. These antibodies can be used instead of antibiotics for the treatment of this. Next slide. Similarly, another project like for Helicobacter pylori, the global population, about 50% of global population are infected with Helicobacter pylori. The Helicobacter pylori not only infection, it can untreated or many persons will develop a cancer. So it becomes very important. So it can, the Helicobacter pylori causes ulcer. If it is untreated, all these complications will get it. This can be treated with antibiotics, but there are 20% of population or Helicobacter pylori are resistant to clarithromycin. Clarithromycin is supposed to be drug of choice. Hence, we need to, alternatively, we need to have some, like any products. We have proposed this antibodies. We got funding for this. So what we can do, we like this, it's an antibodies or again, it's from eggs. Not only since it's from egg, it's a functional food. We can incorporate into even fortify with anything, any probiotics, or simply can curd also can be used for this purpose. It's an ongoing project which we are working with PG Medical College, Department of Microbiology, or a collaborator for us. And another one is rotavirus. We have raised antibodies against rotavirus. It is an alternative to ELISA. We wanted to, using biosensor method, we wanted to detect this virus from the fecal sample, so that the children who are suffering with infection can be treated with antibodies. That's the idea. So finally, we have come to highly purified antibodies. That's the BEG grant we got it, which is kind of cheaper and highly purified antibodies without affecting the quality of egg components like yolk, white. Only the antibodies alone will be taken and will be we'll put it back to the food chain. That's the idea. And the final one, last one, is on coronavirus. This I'll explain it a little late. Next slide. So these are all the products like antibodies, antivenom. We have found some solutions for streptococcal infection which causes dental caries. For rotavirus, we have generated biosensor diagnostic method. For management of dandruff, we raised antibodies. We have made some formulations. For poultry infections like salmonella, we have prevented some horizontal transfer to the human being from the poultry. Poultry is the source of infection. Once if we can able to control this infection, we can control it human also. That's the concept. We can get some clean meat, clean eggs for the export market. It will be useful. And diagnosis as well as treatment of mastitis, rabies. It is for the passive therapy for the basic rabies, and ongoing SARS-CoV-2 and Helicobacter pylori. These two work, which ongoing work we are working on. So for my topic, like the idea, what I have presented it, all this, we got some idea. We made some protocols and we have submitted to the government funding agencies. Initially, we were not about 5-6 times or almost 10 years, we didn't get funds. Later on, we were able to get funds because of our novelty as well as simplicity in the work. When we replace them, like techniques or technology, we are able to get funding. For that, I can give some example. One example is the best example what we have recently got the funding from VRNS. Okay, Bob Atomic Research Center. It's again, when 2019, end of 2019 or 20 January, we got COVID too. So at the time, we have no awareness about this coronavirus. Being in the microbiology department, I have taken some references, collected a lot of references. With our knowledge, monoclonal antibodies, antibody knowledge, we have published a paper on monoclonal, polyclonal, how we can able to use these antibodies and also to make a consortium to prevent this infection as well as to treat this preventive COVID as well as treating. Suppose we are getting infection also. That is how our publication we made it. So it was in a review paper. With that review concept, when the DBT, like many funding agents, they wanted to fund for COVID. That was the concept note they were asking. We made on the concept note. That was the proposal. This is the, I should thank Dr. Ramanathan, Dr. Anand Kumar, and in association with them, I have submitted a proposal because it's like this is a viral infection. You cannot, it's our laboratory may not be enough. Single laboratory, we cannot be able to carry forward this work. Dr. Ramanathan has got cell culture facility, and Dr. Anand Kumar, Indian Immunology, it's going to be like a 3,000 crore industry, that is turnover they have. And viral infections are, they can able to very easily, and they have the facility of class 4 type of viral handling. Any type of virus they can handle. These are 3 facilities they had. With that, these facilities, when we presented, we were able to get funding. I will tell you the objectives we made is like with our experience, as I told you, I learned that 5 objectives. We have prepared the objectives. Okay, objectives 1, 2, 3, 4, 5. It's a content like we wanted to raise antibodies against SARS-CoV-2 spike protein. For that, immunization protocols is there. Then you need to characterize them. Then in vivo and in vitro neutralization studies are required. So in vivo and in vitro neutralization, we need to go for class 3, BSL 3 laboratory, that is Indian Immunology. I said like we will be taken to that. And finally, what you do, that's the question that comes. So these antibodies can be useful for international spray, intranasal spray, where the virus is largest. Or we can able to make a lozenges, keep it in the mouth, so that the viruses through mouth, when it is taken inside, they will be neutralized. Or if they are infected, we can use these antibodies for parental route of treatment for the SARS-CoV infection. That's the idea we got. Today it's commercially tested, it's available. There are companies abroad, some foreign US, they prepared these antibodies already. It's a consortium is available against viruses. We are yet to complete this. Once it comes, Dr. Ashok is working on this, and once it comes, we'll also be able to take it forward. Very important. This slide is an important slide for you, those who are interested in applying or preparing methodology. The clarity in what we got an idea. This is actually the proposed validation testing work plan. That is the assets. I took some slide from the earlier preparation. This slide explains that the concept note. This is the concept we wanted to work on. One side, a polyclonal antibody will be produced. Another side, monoclonal antibody will be produced. Once you can able to produce this monoclonal antibody, what you will do? What are all the characterization we are going to do? Some SDSP, Western blot analysis, flow cytometry. Then that SCFE fragments, what you will be, clinical trials will do it. That consortium will be using it for the preparation of preparation of lozenges or mouthwash or nasal spray. Other side, which will be other side, polyclonal antibodies also we will be able to do it. Suppose monoclonal we are not able to get, polyclonal definitely will get. That is the idea we have proposed. This is the concept note we gave. But they were asking, finally what they were asking is like, once the concept note they saw, they were asking us to give a clear idea what exactly you're going to do. So that's how we were able to prepare that from that, and we got the funding. So novelty statement and concept. These are some of the heading they will give, like when you go for a presentation. Novelty statement and concept note. Once you need to convince them, and also we love to prepare a proposal, workable proposal, reproducible proposals, and which you need to have some alternatives. Suppose you are working, preparing. So later on, suppose you are not able to take it forward, you should have some alternatives. For example, if it is you are trying to clone, and if you are not able to clone it, and alternatively there are some proteins available. With proteins, we can able to take it forward. Commercially available proteins also you can construct this work, and you can raise antibodies work with that. That's the idea. So for this is the paper what we have prepared. Hodge display, SCFP monoclonal antibody, library construction. This is the concept only that we have not done this work, but it is possible. Luckily, we had Ashok had an experience with this because he worked in National University of Singapore. With this concept, we were able to work with this. He has joined in this project. He is working. A smaller size of SCFP antibody molecules holds greater advantage and higher yielding of specific antibodies that can be able to even cross blood-brain barrier, and therefore it can be used as international spray, lozenges. In parental route also, this antibodies can be used. That is a potential advantage of this. This is also again very important alternative to whole antibodies, like mammalian antibodies. Okay. Even this concept can be used for the treatment of snake bite also. It's a growing field. This is another methodology which we thought of, like this purely we got it from the published articles. We wanted to raise antibodies against RBD, okay, proteins and a region of spike glycoprotein. So we have sequenced the amino acid structure, amino acid size. We have taken all that. We made a small proposals. This is like, for example, the virus, from the virus, you can cDNA synthesis you can make with mRNA, single-stranded DNA, complementary DNA. You can be able to RBD spike proteins can be prepared, amplified, cloning, sequencing, transformation, amplification. The recombinant plasmid has been transferred to either E. coli or Saccharomyces cerevisiae. From that, we were able to synthesize the antibody, synthesize the protein, recombinant protein, and purified proteins have been taken and injected into 21-week-old white leghorn chickens. Antibodies has been raised. Those antibodies we have taken it for characterization using titration of like ELISA method, titration of antibodies, sensitivity, specificity, checked it. Then through immunodiffusion, we found that there is antibodies. And finally, the outcome, what you will do with this antibodies. So conclude this, like you need to take it for neutralization studies. So in vitro and in vivo neutralization studies will have to do it. For this, we need a facility, BSL-3 facility. For that, we have Indian Immunological has come for our rescue. This is the part we need to do it. That's how its proposal is going on now. We have taken some in vitro virus neutralization and in vivo neutralization using mouse model, BALB-C mouse model. We are able to propose. Validation studies also we will have to do it finally. So what will be the outcome? That's by generating the CFP, IgA antibodies, okay, monoclonal antibodies in chicken, preparation of antibody formulations such as international intranasal spray, lozenges, and parental route towards prevention and treatment of SARS-CoV-2 infection in COVID-19 patients. So today we are able to, the pandemic, or the past 2 years pandemic, this proposal was done in 2020, within a short period of like within 3 months. And we are end up your project. So now we have some many we all know, like first the infection was, we don't know, many organs are affected by SARS-CoV-2. Next, pandemic, like first cycle, second pandemic, so second wave, it affected our lower respiratory infections. Many people died of oxygen depletion. Third wave with the Omicron, only upper respiratory infection. Many are not to the death. So that's what the mutation. That is the journey we have undergone. This preparation we made it long back, like 2020 itself. So it is possible now. We can able to understand that with the other countries' contribution, or there are already available antibodies are there. Our antibodies also, once it comes, we can able to prepare a consortium, and we will be taking forward as a product to our nation. That's how the end use plan for development of implementation strategy. That's the idea. We can stop here. This is the idea. Like, see, the virus infects once its replication takes place, and it enters through ECE2. This is the ECE2 receptor. The spike protein binds on that RBD, binds to ECE2. When you have the antibodies against that, when you treat them, or when you use it as a preventive measures, or infected patients when you give, or it can able to prevent the infection, or it not enter the ECE2 receptors. That's the concept of this. And for this, we have prepared a proposal. Successfully, we are able to complete. Successfully, we are able to take it forward also, along with Baba Atomic Research Center. There are some scientists, Dr. Kulkarni, who is a co-investigator for this. Another project with simple concept, we got this project like BIG, Biotechnology Ignition Grant. Here, all these years of experience, what we found out is the antibodies are alternative to mammalian antibodies. The antibodies are functional food, and also these antibodies very easily can use it through passive therapy, through oral route instead. When you go for a parental route, a lot of regulatory. One very important thing is regulations. Regulatory will not permit anything. You cannot use it. It takes about 10-15 years to, like life and death, you can able to use it. Like antivenom products can be used for parental route. When you use it as a supplement, food supplement, feed supplement, because the antibodies are egg origin, and you can take it very easily, it is accepted. One concept is that simply you can able to purify antibodies about more than some 50 to 75 milligrams per yolk egg yolk. That is our experience. When we calculate this to produce some 1 kg, like this 100 grams of antibodies, you need to spend about 5 to 10,000 rupees. That is the concept we have presented. So the experts, when we are presenting this, the experts was very much convinced, but at the same time, lot of discussions were there. How it is possible? Can we able to produce these antibodies with that cheaper? Then it will be very much viable for passive therapy. We can use it. Even fortification of food, we can able to do it. And it can be used as nutraceuticals. That's how they were able to support us. During the presentation, like we had about 3. First, the concept note we gave. For that, they called us for presentation. They gave the comments of applicant addressing concern. Like when it goes to the reviewers, they will give the comments. That is the comment I made it here. So we'll have to correct it and give full proposals with the comments. That is how also we can get funding very easily, because the comments, when you can present it, the reviewers as well as when you are doing presentation, also many comments will be there. When you can able to correct this, you can prepare your protocols as well as the entire proposal model, you can able to get funding easily. That was our idea, like that is our experience, whatever so far with examples, with a few examples I gave you. One best example is like we were for SARS-CoV-2, how we got funding, because it was nationally or internationally, it has become an important area, thrust area we chose, and even without the proof of concept, all other funding we got it through proof of concept. We got funding for SARS-CoV-2 virus without our proof of concept, because it's with our experience. We have presented this, we were able to publish an article, and that article got very much importance, and it was published in elsewhere publication. And with that, with quoting with the publications, we have presented this work, we are able to get funding. So the funding also, like, we were able to take it forward because of with collaboration with myself, with Dr. Ramanathan, here PhD College of Pharmacy, and Indian Immunologicals, and also support from Baba Atomic Research Center, we were able to take it forward. So that's the story. And generally, to today's topic, finally to conclude, it demands a clear statement of the problem. That is what we need to address a problem or choose a problem. Okay, then we can able to get a solution for it, or you can able to manage it, you can able to bring some solutions, you can write a proposal. It requires clear objectives and a plan. Then the existing data, like primary data, or we have our own data, like proof of concept, you would have had some patents, or you would have publications. That's very much important with preliminary data. Using both positive and negative findings as proof of concept, you can able to go forward, take it forward. So one thing I would like to say here, like even MSc projects, what our students have done with small funding from Tamil Nadu State Council for Science and Technology. At that time, about 25 years back, they'll give only 5,000 rupees for the project. That 5,000 rupees also was useful. That 5,000 rupees, we established some proof of concept. Those concept was accepted by DBT or BIRAC. We were able to get some 50 lakhs. It has taken to that level because we had some proof of concept. We have good institutions, like not only good, our PhD institutions have got Arts and Science Medical College, Institute of Advanced Studies, PhD College of Pharmacy. Various facilities are available. With collaboration, you are able to take it forward when you work. That's how. With alone, in our arts and science facility alone, to a certain extent, we can get funding. But when you go for some collaborative work, it is easy to get funding. Convincing the funding agents also easy. So the new data should be systematically collected, analyzed to answer, and the original research questions, objectives, when you have with you, it's going for funding is easy. It's essential if the study involves research on human subject or on experimental animals, in order to get the institutions ethical approval and for funding agency. So the protocol preparation is again tricky. Simply, you need to make a concept note. With that, you can able to take forward anything. Even many cancer research in India, antibodies, we have next proposal is like very easily you can take it forward. Cancer antigens, using the antibodies, we can able to neutralize them. That's the concept of SLAC. And we can able to take it forward. Anybody is interested in our group, you are in this audience or teachers, you can work on very easily. You can able to get funding. With this, I thank the organizers for the opportunity. And there are questions, I would be happy to answer for the same. Thank you.
Thank you, Dr. Michael, sir, for your valuable session. The next session will be start at 11:20 itself. So all the delegates, please join at the sharp 11:20 for the next session. Thank you.